It's myself Jagadish Kumar Kara, lecturer in zoology, KBDAB Colony, Rakarpur. Today I am going to discuss about amniocentesis. Amniocentesis is a procedure in which amniotic fluid is removed from the uterus for testing or treatment. Amniotic fluid is the fluid that surrounds and protects a baby during pregnancy. This fluid is present in a sac called amniotic sac. This fluid contains fetal cells and various proteins. These cells and proteins are used to test the genetic abnormalities or infectious diseases is present in the fetus. Amniocentesis is also known as amniotic fluid test or amnio. Let us go to the procedure in amniocentesis. In amniocentesis, a small amount of amniotic fluid which contain fetal tissues is sampled from the amniotic sac. Then the fluid is analyzed in the laboratory to find out the presence of genetic abnormalities or infectious disease whether present in the fetus. Specifically, amniocentesis is carried out to examine the DNA of the fetus in order to find out genetic abnormalities. This is the diagram of amniocentesis. In this diagram, you find that the fetus is present inside the uterus. It is surrounded by the amniotic sac, which is filled with amniotic fluid. This fluid protects the fetus from shock and injury. While carrying out amniocentesis, an ultrasound evaluation is carried out to know the fetal movement, fetal position and placental position. Later, with the help of a syringe, the amniotic fluid is drawn from the uterus. Then the fluid is taken from the syringe and tested in the laboratory. Let us go to the step-by-step procedure. The first step is ultrasound evaluation. The ultrasound evaluation is carried out to determine the viability and abnormalities of the fetus. It also helps to determine the location of the placenta, fetal position and fetal movements. It also helps to know the characteristic of the amniotic fluid. These informations are utilized to determine the type of needle to be used and the procedure to be followed in carrying out the test. Preparation is the second step in amniocentesis. During preparation, the abdomen is treated and cleaned with antiseptic before the procedure. These measures are taken to reduce the chances of infection. The tools used in the procedure are coated with heparin. The heparin is an anticoagulant. It prevents the clotting of blood at the site of injury. Then the third step is needle insertion. With the aid of ultrasound guidance, a needle is inserted through the mother's abdominal wall, then through the wall of the uterus, and finally into the amniotic sac. The area of amniotic sac from the fetus is punctured, and approximately 20 milliliter of amniotic fluid is extracted. This procedure can be done with a single needle or a double needle techniques. The preference of a needle is based on the individualized patient factor and the preference of the physician. After that, the laboratory testing is conducted. The collected amniotic fluid is then submitted for laboratory testing for chromosomal abnormalities. Later, the puncture site heals with time. Then when this amniocentesis is conducted, an amniocentesis is performed when a woman is between 15 and 20 weeks of pregnancy. Women having risk for genetic and chromosomal problem in the fetus generally prefer it. That means this amniocentesis is specifically test for the genetic abnormalities present in the fetus, and this process helps in the management of the pregnancy. Then what are the types of amniocentesis? On the basis of the type of test, amniocentesis is of two types. One is genetic amniocentesis. Genetic amniocentesis involves taking a sample of amniotic fluid and testing it for genetic abnormalities. As for example, for Down syndrome, amniocentesis can be carried out. It provides information about baby's genetic makeup. Generally, it is done when the test result might have a significant impact on the management of a pregnancy or individual desires to continue the pregnancy. It is usually done between 15 and 20 weeks of pregnancy. The second type of test is fetal long maturity amniocentesis. Fetal long maturity testing involves taking a sample of amniotic fluid and testing it to determine whether a baby's lungs are mature enough to birth. It determines whether a baby's lungs are ready for birth. It is usually done between 32 and 39 weeks of pregnancy, because earlier than 32 weeks a baby's lungs are unlikely to be fully developed. Then why it is done? The amniocentesis is done to test genetic abnormalities, to test long maturity in the fetus. It is used to diagnosis of fetal infection. Occasionally, amniocentesis is used to evaluate a baby's for infection or other illness. Amniocentesis is also done for the treatment of the pregnant women. It is used to drain excess amniotic fluid from the uterus. The presence of excess of amniotic fluid is known as polyhydramnions. But it is also used for paternity testing. Amniocentesis can collect DNA from the fetus, and that can be compared to DNA from the potential father. Amniocentesis is also used for sex determination, but nowadays sex determination is legally banned. What are the potential risks of amniocentesis? Amniocentesis is performed between the 15th and 20th week of a pregnancy. Performing the test earlier may result in fetal injury. This test carries a risk of miscarriage. However, amniocentesis is very much useful in the diagnosis of chromosomal diseases and in the management of pregnancy.